Hey YouTube, this is my first video on the channel, MedCrammer. I'm Yusuf, and today we're going to be talking about a very medically significant bacteria called H. pylori, or which is short for Helicobacter pylori. As you can see here, Helicobacter is the genus and pylori is the species. Now this type of bacteria is it is not found in the normal flora or the microbiota of the human. So it is pathogenic to humans, which is why we studied in medicine. And let's start. So first, we want to talk about what is H. pylori. As we said, it is a type of bacteria. Now, as you can see here from this little vector diagram I drew of it, you can see that it has these flagella, and they are on one side only. Okay, so let's look at this here. This is an I think it's an electron micrograph of the bacteria. So you can see here that first of all, it is gram-negative and it is spiral shaped. Now, gram-negative refers to the staining. So typically, when we produce cultures for bacteria, we can use staining to define the properties of a bacteria, which helps in identifying the bacteria. So it is a gram-negative. It stains negative for a gram stain. It is spiral shaped. Now, it's not very visible here because it just looks like a log, but it's basically in real life, if you actually zoom in, it's gonna be spiral shaped, and you can see that it has multiple flagella. Usually they're 5, and they're kind of sporadic here in this diagram, but you can see that they all originate from one pole. So they're unipolar and it's multiflagellated. Okay, and we said that it is pathogenic to humans, which is why we study it. And most importantly, it lives deep in the stomach mucosa. Now, this is very important because we're gonna look at the pathogenesis and transmission of the bacteria. But basically, it lives in the stomach, but not on the epithelial layer, because as you might know already, the stomach has an acidic pH of 2 to 3, and this pH is too low for the bacteria to survive. But deep in the tissue, the pH is more normal, towards the 7, which is where it is found, and it also has mechanisms to counter the acidity of the stomach, and we're going to talk about that. So, in short, that is what H. pylori is. Now, let's talk about the diseases. Now, obviously, we said it lives in the stomach, so you can probably guess what happens. It does. You can see here, it causes a few different diseases, and not all of them are on the same level. I mean, some of them are in pretty chronic and severe cases, and some of them are acute, more mild. So they're not all the same, but they're all related to the pathogenesis of the bacteria. Now, first of all, gastritis. Now, this happens because the bacteria starts to cause tissue damage by multiple mechanisms, but generally it causes inflammation. So this inflammation happens, and then stomach inflammation is obviously gastritis. This can lead to many different complications, such as gastric adenocarcinoma, and in really rare cases, mole lymphoma, and it can also cause peptic ulcer disease. Well, to understand how urease helps the bacteria survive, let's take a look at the urease reaction. Okay, so we know that urease is an enzyme from the name, from the suffix ase. It is an enzyme. Okay, so this bacteria has a urease enzyme. Now, the first part of the urease, so it's related to urea. Okay, so as you can see in the reaction here, urease helps convert urea that is present in the cells, obviously, plus water, so urea and water, in the presence of urease, which is the enzyme, converts it to carbon dioxide, water, and ammonia. Now, carbon dioxide and water are just waste products, but the main focus here is this ammonia. Now, ammonia, if you studied chemistry, is alkaline, which means when an alkaline, such as ammonia, reacts with an alkaline, or also known as a base, reacts with an acid, it neutralizes the acid. So the acidity of the stomach comes from the HCl, so hydrochloric acid. So ammonia reacts with the hydrochloric acid to neutralize the acid, which in this case increases the pH of the mucosa from the normal 2 to 3 to more of a sustainable level for H. pylori. So this is how urease helps the bacteria survive in the stomach. Now the second pathogenesis mechanism, it also can attach to the epithelium with certain molecules that it secretes. I'm not going to go into super detail. This is supposed to be a brief introductory video, but there are molecules called adhesins that the bacteria can secrete, and these help the bacteria attach to the epithelium, so they're not, they're not washed away, or because the stomach does a lot of churning and it has a lot of motility, so the bacteria can easily get detached without these adhesins, basically. And then third, it produces virulence factors. Again, I'm not going to go into detail, but I'm going to leave the sources, so if you want to know more about these, you can read from the textbooks that I study from. So this combination, in the end, leads to inflammation and tissue damage. Now, we talked earlier about the diseases that it can cause. So this pathogenesis is actually related to the diseases that we talked about earlier. So the pathogenesis leads to complications and clinical manifestations, which, as we said, gastritis, PUD, and gastric adenocarcinoma. So this is how it all links up. Okay, now let's talk about the diagnosis. Now, there are two methods, not two methods, but two categories. Some are invasive, and some are non-invasive. Now, invasive include taking samples from the stomach tissue itself. So, like a biopsy, you can take a punch biopsy of the stomach tissue and diagnose it for H. pylori. There's also a rapid urease test, because, as we said, the bacteria has urease. And then you can do a culture for the bacteria and PCR polymerase chain reaction, or the antibiotic resistance testing. That's for the invasive. Now, I want to make other videos explaining each of these methods because I know going over them rather quickly, but this is again an introduction. Now for the non-invasive, you have a urea breath test, a stool antigen test, and serological, which can detect antibodies, but it's not useful for current infection. It's more useful for past infections. Okay, last but not least, we have the treatment. So for many of the bacteria that affect the gastrointestinal tract, we can use first-line therapy, which is a triple therapy. This includes a proton pump inhibitor, which helps neutralize the acidity of the stomach, and then clarithromycin, amoxicillin, and metronidazole. For the quadruple therapy, this is given if the first-line therapy was not very successful, if there's resistance. Again, PPI, and then we have bismuth, which is supposed to enhance the mucosal defense against the acidity of the stomach. And again, tetracycline and metronidazole. So this is it. This was a quick overview of H. pylori. Now each segment here, each of these segments, the treatment, the pathogenesis, each one I can make a separate video on because there's just so much to talk about. But I just wanted to give you an overview of H. pylori. So yeah, if you benefited, please like the video and subscribe, and I'll see you in the next one.